My research basically centers around trying to understand how the cells of our body show to the immune system that they're infected with a virus or other pathogen. I started during my PhD actually looking at adverse drug reactions, so slightly different, but these particular sort of adverse drug reactions were stimulating an immune response that was very much like an inappropriate stimulation of a response against a virus. And so I was trying to understand how the drug was interacting with this system and sort of making a cell look like it was pathogen infected or virus infected. And so that brought about or introduced me to a whole range of techniques which were mapping what's being seen by the immune system on the surface of the cell being presented by HLA molecules. The Michelson Prize gives me an opportunity to gain a little bit more independence and also to investigate a few areas that we didn't already have funding for with regards to some novel insights in how our cells are actually showing that they're infected with viruses. At the moment, we're learning more and more about how the immune system works, and we keep discovering new things, spliced peptides being one of them. All of these discoveries are basically increasing our understanding of how our body shows infection and how we can harness that to stimulate the best immune response. And that's intrinsic to being able to basically generate an effective vaccine. So I've been working with Tony Purcell since during my PhD. So I first started working with him at that point, and then at the end of my PhD, I came over and started working with Tony full-time at Monash University. Tony's incredibly supportive as a mentor and as a boss. He allows us freedom within the laboratory to explore what we're interested in. Tony's worked very hard to get a team of people with very complementary skills. So we have people that have expertise in bioinformatics, proteomics, T-cell immunology, and all of us work together. And so he's very encouraging to collaborations within the group and also without the group. I've always enjoyed learning. That's why I'm here. I couldn't ever anticipate the thought of not doing something where I was learning something new. And science is the perfect place for that. And it also has elements of creativity. Every time you're trying to answer a new question, you've got to think about the way in which you can pose it in order to get an answer that's actually informative. And I really enjoy that process.